draw this out so that we have a different way to look at this. All right, so Griffith took mouse, really crappy mouse, add smooth, mouse is dead. Oh, it's just getting worse. Okay. Take rough. They're happy because they didn't die. Okay. Take heat fix smooth. So they're looking more like elephants or horses as they go along, aren't they? So smooth, heat fixed, or heat killed. And anybody recall what happens with this one? If we kill those smooth, those ones that are capable of forming capsules, do they cause death? They need to be alive to actually cause the infection. So these mice come out looking fine. And then the last of the experiments here was take a mouse, take rough plus the heat fixed or heat killed smooth. And somehow now these mice are dead, or they die in greater frequency. And when he took a look at the colonies on the plate, there were some rough colonies and there were some nice smooth colonies as well. Something that we wouldn't expect, given that the only cells that were alive here were the rough ones when they went into those mice. And what I pointed out was that this is a model of transformation. So the question, if we go back to that original question there, is explain how transformation led to these observed results. Okay? And one of the things that I kind of alluded to early on was just saying, if you can explain transformation or the process of transformation in general, that's going to get you some points. That's kind of a good answer. And where you go from a good to an excellent or great answer on this is really in the context of how do you put this into the context of this particular experiment. So using some of the terminology or evidence from the experiment to support this model of transformation. So let's just start with just a really quick review. What is transformation? Ashley. Good. Picks up DNA from a lice cell. In this particular case, so to go to the level deeper then, which of these is the lysed cell? Smooth. And those that are picking it up or being transformed would be the rough, right? What else is true about transformation? So after that DNA has come in, what has to happen? What was that? Recombination. And recombination requires what? Between the piece of DNA that's coming in and the genome requires what? Homology or similarity in the sequence somewhere? And so these rough cells, they had the gene encoding for a capsule, but it was mutated in a way so that they actually, even though the gene was there, they couldn't create the capsule protein. So a homologous recombination or recombination event had to have occurred to allow for that gene allowing for smooth to come into the genome and flip out or excise or remove the mutated version. Okay? So other things that you would be looking for in a good response or in a better response here would be things like recombination, homology, to describe that those two pieces of DNA are being swapped. And then the last part here is just putting on the fine detail. That it's the smooth, the DNA from the smooth cells that's coming in and transforming those rough into smooth colonies. Is it 100%? No, and that's not what Griffith observed. It's not 100% of the time that this is going to happen. It's a rare event. And I think one of the things somebody asked on Wednesday, I can't remember who came up at the end of class and said, hey, so the transduction picture makes it look like it happens like 1 out of every 7 times. Is that right? And that's absolutely not true. Every single one of these events is quite rare. We're talking on the order of transduction of a single gene, we're talking on the order of maybe 1 out of every 100 million bacteriophage that get made might be a transducing phage, one that's able to transfer genes from one organism to another. Conjugation is rare. So is transformation. These are all rare events. Oftentimes with transformation, that DNA comes into the cell and it gets chopped up into individual nucleotides that the cell can use to support its own replication. And so the reason why we see them, though, is because bacteria do repopulate so quickly. But in the context of how frequently it occurs, we're talking about things that happen 1 every 100 million, 1 every billion times, so quite rare events. If we were to put that into human context, we'd say that this would mean probably like if humans could transduce, for example. Given the population of the human population, we're talking like 7 to 70 people on the face of the earth would have some sort of transducing event. That's kind of to put it in the context of people. Based on that, we have, what, 7 billion on the face of the earth, roughly, I think? Okay. Okay, so 7.6 people, then, would be transduced. Any questions on this one? Or any questions on what I'm looking for with a response on this? Okay, so again, this is up. It's under the molecular biology, that last one. So let's move into viruses, and this is kind of a, gosh, you know, when we talk about microbiology, there's so much that we could cover, but there's so little that we really do cover. We focus so much on bacteria. There are fungal infections that we could consider part of microbiology. There are parasitic infections, viral infections, and we're really just going to take 3 days to talk about this. We're only going to take 1 to talk about human. Are a classical model of how viruses work. We actually know more about bacteriophages than we do about most human viruses. And so it's a really good model for just, in general, what happens over the course of a viral infection. What are the steps of that infection? The other reason why I think that this is a really interesting topic to look at is because within the past 10, 15 years, there's really been a reemergence of studying bacteriophages in the context of human health. So let me kind of set up the scenario here. 1920s, bacteriophage were first discovered by a scientist, French scientist named Félix d'Herelle. He ended up thinking these would be really good to treat cholera infections, and he actually successfully used them to treat cholera infections in India. 1940s come around, penicillin is discovered, end of bacteriophage as being potentially used for human health to reduce bacterial infections. Penicillin and the antibiotics became the drug of choice in the westernized nations. In Eastern Europe, Russia in particular, they use bacteriophage nowadays to treat skin infections. That's where I think we're going, or where we're coming back to in the United States, is phage have been looked at as kind of like an alternative to antibiotics. These are actually being used to treat foods. So there are some companies, the brand name is Listex, what they end up doing is using bacteriophages as a way to reduce the concentration of Listeria monocytogenes in food items so that they are safe, ready-to-eat foods. Okay? So this is being used now, and I think that it's just kind of an important way to get the idea that yes, we talk about kind of these traditional things with microbiology, but this is evolving science as well at the same time. The majority of what I wanted to spend time on today was just kind of walking through the life cycle and talking about the different parts of this particular set of viruses. And with all your pictures, you saw that this kind of looks like we've got our capsid, or our head, we've got our sheath, tail fibers. I described this as like that little toy in Toy Story. I have a friend that describes these as ghost lollipops or ghost Tootsie Roll Pops when you look at them under an electron microscope. because here is the lollipop part, and here is the stick of the lollipop. Okay, this is kind of the traditional structure of what we think of when we think of bacteriophages. There are others, but this is kind of just the traditional structure. What is the function of this viral structure? So if you were to summarize, and this might be based off of your descriptions that you have, what the purpose of this structure is, how would you describe that? What I want to do is I want to give you guys a minute to think about that. So look at your descriptions, look at what these different parts are, and maybe you can coalesce around an idea or two of what is the purpose of this thing? Okay? Not what's the purpose of a virus, but what's the purpose of this actual structure? Okay? I'll give you a minute to think about it, and feel free to talk with someone else. Comments that I heard real quick. So one of them was, wait, are these good or are these bad? It's a matter of context. It really is a matter of context here. If you're talking about using bacteriophage to reduce bacterial numbers, that's a good thing. But in the context of transduction, possibly spreading antibiotic resistance through horizontal gene transfer, we view that as a bad thing, right? And a bacteria would view that as a good thing. It's a matter of context here. Okay, so I don't know if it's really necessary to define it as good or bad or however you want to define it. It's just, it is what it is, to be very philosophical for a moment. The second question is, I want to point out here that bacteriophage, these only infect bacteria. And it's not as if there is a single bacteriophage that infects every single bacteria. It's very similar to how how do I want to describe this? It's species specific. So in the same way that there are viruses that infect us, and then there are viruses that infect our dogs or cats or turtles, but those viruses that infect them don't infect us, the same thing is true for bacteriophages. They don't infect us, they infect specific species. And they can even infect specific strains. So what do I mean by that? One of the areas, again, where this is thought to be used, or where folks are thinking of using bacteriophages, is as an alternative to antibiotics. One of the things we'll come back to on Monday is, again, talking about the downside and upside of broad and narrow spectrum antibiotics. Broad spectrum antibiotics can wipe out the natural microflora in your gut if it's an oral antibiotic. Bacteriophage, we can find bacteriophage even with just in the water samples of viruses that infect pathogenic E. coli, but don't do anything to the normal E. coli within our gut. Okay, so it can be that specific, and that's really a powerful tool. That's where the folks are thinking how we can use this. Okay? So getting back to the structure then, what is the function of this structure? And you may have a couple of parts to it, but overall I think we can unify this. Kaylin. Yeah, so what actually helps this bacteriophage attach to a bacterial cell? Good. Those tails are really kind of the initial source of contact there. And what they attach to are specific patterns or genes on the surface of these bacterial cells. So it might be a porin protein, for example, or it might be there are some viruses that attach to the lipopolysaccharide. There are some that attach to flagella. So depending on what it is, that will change the location of where this bacteriophage binds. And then, as you mentioned, once it binds, we've got our DNA here in that capsid or that head. And what essentially is happening, and this is really cool, there have been some studies to look at how do these tails fibers change as they bind to a bacterial cell. And what happens is you basically have an interaction where they kind of sit and then all of a sudden it flattens. So it's almost as if like those tail fibers contract or break a little bit. And what that phage does is it pushes its base plate into the membrane. What does that base plate have on the end of it? Yeah, it has lysozyme and it has other lysins or genes that are involved, proteins, that are involved in degrading or hydrolyzing that peptidoglycan. And so at that point, as it starts to hydrolyze or break apart that peptidoglycan, now that creates essentially a space for a contractile tube to insert itself through that peptidoglycan, through the outer membrane of a gram-negative, through the inner membrane, and now, and this is thought to occur just passively, that DNA makes its way into the cell. Carla.
For the overall
Yeah.
function of it, can we say that it is effectively?
Oh, yes.
Never,
Absolutely. I think that is the other part of this. So one, I think this viral particle here, it's a delivery system to deliver the DNA. The other part is the protection. Because DNA in the environment doesn't necessarily last a whole long. There are going to be nucleases secreted by bacterial cells that may end up destroying that DNA. And so it's kind of an end of the road for the organism. Absolutely, it's to protect the DNA. That is definitely the other role for it. Any other ones? I think those are the two major, at least how I think of it. Okay. I want to point out here, too, that the DNA is the only thing that makes it into the cell. Everything else remains outside. So how do we go from DNA to, at the end of this life cycle, producing more viral particles? What drives that synthesis? Kaylin?
it's a guess, but because the bacteria replicates and so it has part of that DNA from the virus.
Say that again?
Because when the DNA goes into the bacteria and the bacteria replicates to more bacteria, so that's the way it spreads.
It does play off of that. So let's walk through replication again real quick. What enzyme is involved in replication? DNA polymerase. 96%, the vast majority of bacteriophage genomes are DNA genomes. They get replicated by the DNA polymerase from the bacterial cells. So one hallmark that's true about viruses, and Kathleen will talk about this too in the context of some of the others, is that phages and viruses are about utility. They only carry the genes that are essential to their life. Most bacteriophage don't carry along, into the cell at least, a DNA polymerase. They may have one in their genome, but early on, the replication is going to be driven by the bacterial cell, and also transcription will as well. Transcription will be driven by the bacterial cell to start making mRNA. And then what's going to translate that mRNA? The bacterial ribosome will translate that mRNA. In other words, the bacterial cell gets the life cycle of the virus going. Okay? I'm just going to move ahead real quick because I want to... See, these are the ghost lollipops. I just wanted to make sure I point out the ghost lollipop real quick. Okay. I want to put this into the context of what we've talked about so far, so I'm going to just focus on this slide real quick. We've talked about the attachment step, the attachment of those tail fibers to the host cell, the penetration step, or in eukaryotic viruses, we call this an entry step. This is where that DNA is now making it into the cell. And then we get, third, biosynthesis. Replication of the genome, production of mRNAs, production of viral proteins. And those viral proteins just kind of, they come together, they self-assemble. There's some really great videos on YouTube, one where this guy has basically like 10 faces of a ball all in separate pieces, but they all have magnets on them. what he ends up doing, he puts them into a jar and starts shaking them and shows how they assemble together to eventually create a ball. And that's kind of what happens here. Just by random movement, you start to get the formation of these bacteriophage heads, because instead of magnets, we have protein-protein interactions that are driving it. But this is completely random, the creation of these bacteriophages. And it's random in the sense that it just requires movement of those proteins. It's non-random in that they always, or most always, form a bacteriophage structure at the end. So there are specific attachments driving that synthesis. Now, I've given you guys kind of a worksheet because I want to kind of walk through these various steps. You do not need to write down every single thing on here, but this might be one way for us to think about this life cycle. And then we're going to talk about how did the virus exit from this as well. So we've talked about so far that DNA making its way into the cell. Here's our attachment step and penetration step kind of combined into this first image here. So make sure that I write on this. All right, so that's our attachment and penetration. And then the next step here, what we find is those phage genomes typically will circularize. So they usually have sequences at the end that allow that genome to circularize, so that this is kind of like a mini plasmid or a mini genome inside the bacterial genome. To give you a sense of size, again, I've been using the E. coli genome size of about 5 million base pairs as reference throughout the semester. Phage genomes are usually about 100,000 base pairs. That's kind of an average. There are many that are smaller, many that are maybe twice the size. But on average, the phage genome is about 1/50th the size of the bacterial genome. And so that already tells you that phages are going to carry fewer genes. They're only carrying the ones that are required to make the capsid, to control the bacterial cell in a way that promotes viral replication. And basically, the goal of any virus is to turn the bacterial cell or the human cell into a factory to make more virus. I do not watch The Walking Dead, but if you watch The Walking Dead, basically viruses kind of turn those cells into zombies. So they control the function of the cell. We'll come back to the lysogenic in a minute. I want to just focus on the lytic. And lytic here really refers to the virus gets in, replicates, gets out as soon as possible. We're talking 40 minutes. A bacteriophage can go through its life cycle in E. coli within 40 minutes. And from one bacterial cell, you can get, from one phage infecting one bacterial cell, you can get out 100 new viral particles. It's a massive explosion. 100-fold increase with just one cell being infected. So after that biosynthesis, we get what we call the assembly phase. In that assembly phase, now this is where that random movement of those proteins is going to create new bacteriophage particles. Viruses have mechanisms to ensure that their DNA is going to make its way back into the capsid. We're not going to get into the detail of that. But again, this is where we can see mistakes. We talked about transduction. That is the step where we see transduction occurring, in which, by mistake, a bacteriophage might take, instead of its own genome, might take up part of the bacterial genome. The reason why I can do this is that bacteriophage, towards this later portion of their life cycle, they actually chop up the host DNA into small pieces. They start to degrade it or break it down. So that ensures that that host cell is going to die at the end of this process. In the last step here, this is exit. Some folks, the term that sometimes gets used is egress. We'll just call it exit here. So in this exit step, the virus, of course, releases from the cell. They don't release very kindly, though. They rupture the cell. And rupturing the cell requires a breakdown of the peptidoglycan. And not only do these bacteriophage have lysozyme on that base plate to allow them to inject that DNA, but during this later part of biosynthesis, or assembly, they're mass producing what we call lysins, which are lysozyme and other proteins like it, that as they are floating around in the cell, eventually come into contact with that cell wall, start to hydrolyze it, weaken it, and these cells just kind of explode out of one end of the bacterial cell. I'm just going to go. Oh, I don't have a. Oh, yeah, there we go. There's the picture of it. So this is one example here of a bacillus, or what was a bacillus. And you can see how all of these phage, all of these little dots here, represent bacteriophage, and they've just kind of ruptured from one end or one pole of this bacterial cell. I mean, we should have done this lecture on Halloween. This is the stuff that like nightmares are made of, right? No? All right. Okay, so this is just kind of the overall general process. Again, that's lytic life cycle. Goal is to get in, get out as quick as possible. This is in contrast to lysogeny. What is lysogeny? Yeah, so the bacteriophage DNA instead gets integrated into the host genome, and then as the host genome replicates, the bacteriophage DNA replicates along with it. And this can occur an infinite number of times. To go from this lysogenic back into the lytic life cycle, we need some sort of stress or some sort of environmental cue from the bacterial cell that the DNA is being damaged. So UV light is a way that you can take these lysogenic phage and basically trigger them back into a lytic life cycle. Using what we would consider a carcinogen, something that damages DNA, would do the same sort of thing. Sometimes the phage never make it out. And over enough generations of the bacterial cell, that bacteriophage genome becomes kind of pieces get lost, and so these bacteriophage are no longer able to ever kind of come out of the genome again. We see this time and time again. Actually, we're seeing this kind of information in our own human genomes, where we've got kind of a timeline of history of viruses that have integrated their genome into ours. They just don't produce virus anymore because there's been enough damage or enough nucleotide changes to those viral proteins that they actually can't produce virus anymore. This is another mechanism. I want to point out, this is another mechanism here. This is not transduction. Okay? It's similar to transduction in that there is an integration step. It is different in that it's not a recombination integration. So there doesn't need to be homology between the bacteriophage genome and the host genome. But just like transduction, this can integrate into the bacterial genome antibiotic resistance markers, toxin genes, etc. So this is another form of gene transfer in a way. We don't classify it as horizontal gene transfer. I don't think I covered this slide in the video, and that's because I wanted to take the time to cover it here today. This is kind of a hard concept. So the question is, what's the benefit of these two life cycles? Both of them, actually, you could argue, I don't want to argue that one is better than the other, but there's benefits to both of them. And again, it's in part context dependent. So what I want you to do real quick, apologize for the resolution of this, but I want you to think about what could be the potential benefit of a lytic life cycle or a lysogenic life cycle to a bacteriophage. one of the things that maybe we need to put into context here is the goal of any living organism, if we are to distill this down to the most Darwinian example that we can, is for organisms to pass on their genetic information to the next generation. In the strictest sense of biology, that's the goal. Okay? Think about that and think about how do these two methods contribute to that? So how do these two methods allow for organisms to pass their genetic information on to the next generation? And where might these two different methodologies be beneficial for that? Okay? I'm going to give you guys a couple of minutes to think about this. This is kind of a hard concept, and then we'll talk about it afterward. And feel free to talk with somebody else about it. You can't look at a bacteriophage and say, like, oh, this one's going to happen or that one's going to happen. I take that back. You can look at its genome and determine that. But it's kind of random in some cases, I guess is maybe the best way to describe it. The vast majority, and I should point out, not all bacteriophages, not all, if we want to call them species of bacteriophages, not all of them go through the lysogenic life cycle. All of them do go through the lytic life cycle. Lysogeny is kind of a unique event. And for a bacteriophage that might have a lysogenic life cycle, it may happen 1 out of every 10,000 times it infects a cell. So this figure doesn't really display it all that much, but oh crud. This here happens the vast majority of the time, that lytic life cycle, and lysogeny is a much more rare event. Does that help a little bit? So going back to the question at hand then, what is the potential benefit of this? Why might lysogeny be a good thing? Or why might lytic life cycle be a good thing? Meg, do you have an idea? Okay, so and that's how this one is showing more at one time. And we'll come back to that, okay? I think that this picture may be a little bit misleading, but I like that idea. We'll come back to that. Haley, did you have one? Yeah, the reproduction's super fast, right? We said that you can get 100 bacteriophage out of some of these cells within a 40-minute time span. That's huge. And that occurs again, and in a bulk culture of E. coli, that occurs again and again and again and again. In our lab, so with myself and the students that are working with me, we can get out 10 to the 11th, 10 to the 12th bacteriophage per milliliter in an overnight culture. I mean, typically from an E. coli, we can get bacteria at about 10 to the 9th, 10 to the 10th per milliliter from an overnight culture. So we can get vast amounts of these phage. And that can be a very good thing. Get in, get out, replicate as soon as possible. That's really good when, I'm gonna put this in quotes, nutrients are high, or when your host population is really large. So in a bulk culture, in the human gastrointestinal tract, where concentrations of E. coli are large, a bacteriophage that infects E. coli, it's like a kid in the candy shop. Go in, eat all the candy in one area, get out, go to a new area, eat more candy. That's what those lytic bacteriophage are doing. They're getting in, getting out, and replicating as fast as possible. In times when nutrients or cells are a little bit more rare, this is where lysogeny can be beneficial. So Meg was pointing out that like you get a massive explosion all at one time. One of the things that this picture really doesn't show is these cells don't all go into lysogeny or go back into the lytic cycle synchronously, and they don't all have to be in the same location. Think of how we interact every day with our environment. I have touched this table about 50 times already. I'll then go to my office and probably touch my computer keypad. And then I'm going to go to Janesville tonight and I'm going to probably go touch my TV remote. Each of those contacts leads to the spread of bacteria. And if there's a lysogenic bacteriophage in one of them, that also allows for dissemination or spreading of that bacteriophage genome as well. And at any of those locations, at any given time, there might be some environmental stress that triggers that bacteriophage back into a lytic cycle to allow it to reproduce and then to infect new bacteria in that new area. The benefit of lysogeny is kind of like it's similar in some ways to what we see with viruses like HIV. The cell doesn't really know it's infected, number one. It lives, it replicates. And at any given point, you might get reactivation of the virus. And it allows for it allows for spread of the virus in a covert way. Okay? That's the benefit of lysogeny, is that in cases where the nutrients are low or cells are low, this allows for the virus to be spread over vast areas and allows for it to share its genetic information with the world. Okay. So is one better than the other? No, not necessarily. Both have their benefits. Okay. Well, any last minute questions? All right. Have a great weekend, everyone. I'll see you all on Monday. Yeah.